Hello everyone, myself Janavi Gangwani presenting a video under the guidance of Dr. Tushar Wankhede Sir, Department of Botany from Sri Shivaji Science College, Amravati. The topic is haploid plant production and its significance. The contents are as follows: introduction, history, types, applications, significance, conclusion and its references. Introduction. Haploid plants are characterized by possessing only a single set of chromosomes, gametophytic number of chromosomes, that is N, in the sporophyte. This is in contrast to diploids which contain two sets, that is 2N of chromosomes. Haploid plants are of great significance for the production of homozygous lines, homozygous plants. History. The Indian scientists Guha and Maheshwari in 1964 reported the direct development of haploid embryos and plantlets from the microspores of Datura. Types. There are two types of haploid plant production. First is androgenesis, second is gynogenesis. Gametophytes develop after meiosis both in anthers and in ovule, called as microsporogenesis and megasporogenesis respectively. What is androgenesis? Haploid production occurs through anther or pollen culture and they are referred to as androgenic haploids. Anther culture: flowers, then isolated anther, anther culture, then callus, haploid callus, then haploid plantlet, and then homozygous plant. Now, pollen culture: flowers, pollen isolation, isolated pollen, pollen culture, multinucleated pollen, pollen embryo, then haploid plantlet, and then finally homozygous plant. Now we studied androgenesis, that is anther culture and pollen culture, also called as microsporogenesis. Now what is gynogenesis? Ovary or ovule culture that results in the production of haploids known as gynogenic haploids. Selection of opened or unopened flower, washing, then kept in laminar airflow chamber, surface sterilization, then again washing, separation of ovary in regeneration, incubate the culture for 16 hours at 25 degrees Celsius under fluorescent light. In induction, incubate the culture for 16 hours at 25 degrees Celsius in dark. In both, after 2 weeks, we'll get haploid plant. Now we studied about gynogenesis, that is ovary culture and ovule culture, also called as megasporogenesis. Applications of haploid plant production. Genetic variability are induced, mutagenesis are induced, insect-resistant varieties are developed, disease-resistant varieties are developed, and salt-tolerant varieties are also developed. Now let's see significance of haploid plant production. Haploid cell cultures are useful material for induction of mutations and to study the effect of mutation. This method can overcome the masking effect of presence of dominant gene. The screening method for detection of mutational effect is also easier in this technique. The conclusion obtained is rapid production of homozygous lines, speed up the breeding by fixing of the elite inbreeds, shorten breeding cycle and cost saving. The references are as follows.